mathematics and cancer. He received both his bachelor's and PhD in physics from the University of Barcelona, Spain. From 94 to 96, he was a postdoc at MIT developing MEMS under Marty Schmidt and Mark Wrighton. In 97, he joined Mehmet Toner's lab as a postdoc at Harvard MGH to apply soft lithography to tissue engineering. He's been at Seattle's University of Washington BioE, where he is now a full professor, accumulating over 11,300 citations. In 20 years, he supervised 19 postdocs, 12 PhDs, 15 MS, and 43 undergrads. In 2001, in 2014, he was elected to the AIMBE College of Fellows. He serves on the editorial board of Micro Machines since 2019. He's the author of 5 books, sole author, including Introduction to Biomems, 103 departments in 18 countries, and Microfluidics, Hidden in Plain Sight. Since 2007, the lab runs a celebrated outreach art program called BATE, Bringing Art into Technology, which has produced 7 exhibits, free images related to microfluidics and microfabrication, and a YouTube channel that plays microfluidic videos with music, which accumulated 163 visits since 2009. Without further ado, the floor is yours, Professor Folch.
Thank you. I guess I have to. Good?
Yes.
Thank you so much for the invitation, and I also want to make a bigger thank you to people like you who make the effort of putting together these talks that are now, I guess, have been very, very important since COVID. To disseminate science through the networks, and I think I'm aware that these are watched all throughout the world. So I think that's just been very important. I think this the impact of these talks of this mode of this the talks that we see are going to be. The impact are going to be very important for the future generations who, when I remember how I learned science, I had to go to the library to photocopy papers during my PhD. So I'm very excited about the possibility of reaching out through this mode of communication. So thank you so much to the Terasaki Institute for organizing this. So today I'm going to talk about this work that we've been doing for now more than a decade on tumor-on-a-chip platforms, which allows us to do drug testing with more fidelity, I would say, not only high throughput, but more precision. And this started in roughly in 2012, 2013, and it has been heavily funded by NCI. We've published a number of papers, and we also are in the process. We also put together a patent, and this we could not have been. We would not have done this without a number of collaborators. The clinical collaborators are shown here in yellow at the bottom, but also my present more solid collaborators, Professor Taran Gushral at the Hutch. So what you're going to see today is the body of work of a very large team, not just my lab, and so it all goes in the framework of what we call functional drug testing. Okay, why do we do this? We do this because we are concerned about it's actually a very huge problem that tissue culture models and animal models on which preclinical tests, drug tests are based, which have been, pharma has been testing drugs in these models here, I'm going to use my laser pointer here. So on the left, that's how pharma has been doing their preclinical tests. Do not recapitulate the physiology, the pathophysiology, and the physiology of humans. And so that has been the reason why clinical trials have failed and or are failing. And I'm not the first one to say this, obviously. There are most cancer drugs take about 2.7 billion on this is the median or the average, I forgot, to develop, about more than a decade to approve, to go through the FDA process. And so in the end, less than 4% of cancer drugs get approved during this process. And so about more than half of these failures are due to the lack of efficacy, precisely because there's a mismatch between the physiology and the pathophysiology of these surrogates where the tests are done in the preclinical stage and the patient. And so we envisioned very early on that this would be an opportunity for microfluidics, because these biopsies that are extracted are very small. And so we thought, okay, it's a very difficult problem because the biopsies are small. It's a very difficult problem for most people, but it's an opportunity for microfluidics because we can put a lot of tests into a very small volume or a small area. And so at the time when we started, we didn't know what the design would look like, but I recognized that we had the skills to produce designs to put a lot of fluids into a small area. So that's the essence of what we do. Is summarized in this slide. Is how we want to use microfluidics to do a lot of tests, so we can do the direct testing in with human biopsies and bypass animal testing, so that we can get that efficacy problem solved. Okay. Well, there's been quite a bit of this idea has been attempted by other people. People have been trying to do functional tests with tumor organoids mainly, and this has had some success. And so tumor organoids can recapitulate some of the pathophysiology of the human organs and of the cancers. The main problem for cancers, the tumor organoids, because they are grown, they lose all the immune phenotypes, a lot of the vasculature as well. These are the first type of cells to go because they're grown. And so this for immunotherapy, these are insufficient models. There's been also a lot of efforts in using tumors that are directly microdissected, and here's where we think there's a lot of opportunities for microfluidics, and also the direct slicing of tumors into there's another mode of microdissection into slices, and finally the direct microdelivery of drugs in vivo. So if you order these this can be ordered by throughput. Of course, tumor organoids you can achieve very high throughput because there's a growth process, and at the bottom you get much lower throughput when you do the direct delivery in vivo with needles and so on. But in the middle, there's an opportunity for microfluidics because for these formats of tissue, you can increase the throughput substantially by using microfluidics. So we decided to concentrate on these formats, And here you can still retain the tumor microenvironment to a point where the relevance of the testing is extremely high. Here at the bottom, I would say that there are also concerns for the safety of the patient, and that's why we didn't want to concentrate on that. So the first thing we looked at was the microenvironment of the preservation of the microenvironment of in slices. This is something we did very early on to check that these were indeed suitable models. And they're full of macrophages. This is the top row is day 0, bottom row is day 2 and 3, and they're full of immune cells and endothelial cells. So and they're naturally proliferating. They actually grow in thickness, the slices over time. So they seem to be healthy. And then we put together a platform that you see here. I'll show you in a second how we make it and how it's operated. But essentially, I just want to show you this pretty picture that is, but just to show you that it's very intuitive to operate, because it's just like a multi-well, platform. The sample goes here in the center. And I'll show you how it shows, but it is a single microchannel that goes from each of these wells into these. And I've always believed that microfluidics should be a little bit like the mechanics of a car. It should be hidden from the operator, because it's just a precision mechanics, and the user should not touch it. So that's kind of like the idea behind this. The user is filling these wells and not having access to the microfluidics, which is running underneath and powering the precision of the assay. And then there's one outlet that is common to all of these, and because of the design, they all have the same flow rate. They all have the same resistance. So the way and this is that we actually put the sample with a transwell. I'll show you in a second. So the way it's fabricated, it's all made in plastic. And we designed it. We started the design with a computer, and it's all digitally manufactured. And then we do all either by laser cutting or by CNC milling. And then all these pieces are put together with thermoplastic bonding. It seems easy here, but it's actually very difficult. I just want to give credit to my students who have spent a lot of time doing this. And we insist on using thermoplastics because thermoplastics have the top ratings in biocompatibility, and we cannot compromise on biocompatibility when we do devices for clinical work. So that's why we cannot even think of using PDMS and things like that that have potential leachets or potential problems of manufacturing later on. So we do need thermoplastics. This is just a quick, just to show that there are complexities in dealing with thermoplastics. You have to use bonding using solvents and so on. I'll be glad to give you the details, although they're all in our publications. And what's hard about it is to do it on a large piece of plastic on the area of a 96 well plate. It's fairly easy to do, to bond small pieces of plastic. It's much harder to do it at 100% yield with the area of a multi-well plate. So the way the platform operates is, this is shown in a cross-sectional area, and it's that when the channels have an open area in the middle, it does not work unless you cap it with a porous membrane. That's from the transwell that I showed you earlier. And so the first thing we do is we add the porous membrane, then we fill it with we fill these wells, and then we start pulling with a syringe pump. And at that point, the drugs, the fluids, start diffusing from underneath the porous membrane and feed the tissue slice from underneath. This all inside an incubator at 100% humidity, and so there's no there's no drying of the tissue. It's this mode of feeding tissues in vitro is very old. It's based on organotypic tissue slices, which was pioneered in the 1950s, 1940s. I forgot, and so that is that idea is not ours. What we did is we combined it with microfluidics so that we could have many drugs underneath. We've also combined this with needle core biopsies, and needle cores are essentially out of the clinical standard, and so we've verified that the controls, essentially the histology that we get from the tumors on day 0 is the same as the cores that we have kept on the device up to 4 days, and if you put them down, these cores have a flat end, and we put the flat end on the device, on the membrane. You can make them, you can expose them perpendicularly on the lines of the device, so you can see that you can get selective exposure of a core. So these are very interesting from a clinical standpoint. But in general, what we do is we put slices on this end, on the top thing. We first culture them. The first hours we first culture them on a normal transwell, and then we transfer them onto the center of the device, and that part, just that little bit of suction that created by the outlet as it pumps out creates the needed adhesion this area becomes the roof of the channels, and then by diffusion getting these drugs, in this case these are dyes, so you can visualize the pretty pattern, getting infused into the tissue. We've done a lot of, we always get asked the question, and this was a question in the paper, of course, that we published a while ago, of how much spread do we get, because this diffusive spread from line to line. And so we determined it is very lengthy experiments where what you see here is actually a slice that has been cut like this. So if this were a tissue slice, you take a finger, a piece of a finger, and you put it sideways onto the microscope. You cut a sliver of it, you put it sideways so you can see how much the dye, in this case, one of the dyes is actually a drug, doxorubicin, which is fluorescent, and how much it has penetrated. And we did the experiment twice, one at 4 hours and one at 48 hours to compare. And you can see that at 48 hours, of course, it has penetrated much more, but it has also spread much more. But from these, we were able to determine that we could stop the spread sideways by separating the channels by a buffer channel. So in essence, of the 40 wells of the channel, we have to sacrifice half of them with buffer to separate the other ones. But just one buffer is enough. And then we went on and did experiments with a panel of cancer drugs, and I'll talk about the concentrations a little later but for now just bear with me, and here is it's essentially that these are from mice, so these are very simplified system. These are not from patients yet, and one thing that you see is that they're very homogeneous. So these are not a real tumor. If you could see, and I'll show you in a second, a real tumor is much more heterogeneous. So what you see here is that when you expose these slides here to parthenolide or to cisplatin, the whole slice looks very homogeneous. Okay, that's because xenografts are very homogeneous tumors. They grow in a very homogeneous way. But these allow us to do some research into, especially some characterization of the device. Okay, so these are very good tests. This is from GBM, from a glioblastoma, from an actual patient. And the one thing you can see here immediately is that this is very heterogeneous. So these traces of cisplatin, in some they're taken at different places here, and you can see that some areas respond to cisplatin, some areas don't. That is what you would expect, because in normal tumor, there's areas that have stroma, and there's areas that don't, and there's areas that have more, etc. So that is a challenge with real human tumors, and this is exemplified here perhaps even better, because this is from a colorectal liver metastasis. And we get from our collaborator, Dr. Ray Young. And the good thing about these is that the stroma is visible by fluorescence. It's red fluorescent. So we can see it, and that allows us to quantify it ahead of time. We can see that we can quantify the tumor area. We can see that the areas that have less tumor are also correlated with the areas where there's with the areas with the proliferation area. So you see these correlations, right? And so essentially, what this tells you is that there's areas in which you cannot extract information because there's no tumor, and this is always going to be a challenge. The amount of information that you can extract from tumors is always going to be limited by the heterogeneity of the tumor. This is a fundamental problem. So based on these, on these limitations, we started thinking about another way of creating of extracting information from these tumors, and we came up with an idea of micro-dissecting tumors, and this was in part inspired on the work of Thomas Gervais, who had done some similar work in which they had manually punched some tumors, but we thought we could do better by doing mechanical dissociation, and we essentially used a chopper This thing has a blade that goes up and down, and we set it to go up and down every 400 microns, which is the sizes of the slices that we were used to. And then we set these slices down, like you see here. And then we did another cut every 400 microns, which you see the lines here. And then we turned this dish here side 90 degrees to obtain these 400 micron cuboids. So if you do the math and you think about a cubic centimeter and you cut it every 400 microns, you'll realize that you can obtain more than 15,000 cuboids per cubic centimeter theoretically. We don't obtain that many because we lose material, obviously, but more the reality is perhaps we get about 10,000 cuboids per cubic centimeter. It's rare that we get tumors that big also, but I'm just saying the density is very is that high. But so imagine you can start the number of assays you can start doing with this many cuboids. So we looked into the preservation. The first thing we did is like we did with the slices is we started looking at the preservation of the tumor microbiome. And then the first thing we worried about is was there any damage done by the cutting? And I have to quit here. And we did. We looked at how the internal organization is done by a form of confocal microscopy, and you can see that there's only damage on the outside, like a single layer of cells, but which is expected from the cut, but otherwise the inside is intact. It's so intact that we were able to reconstruct the vasculature, which this opened up another line of inquiry in our lab, which is that, and this kind of I'm doing a little parenthesis here and going a little bit on a tangent, because we started thinking about, what if we could inject some flow into these cuboids, because it's very interesting that as I'm sure solid tumors are poorly vascularized. So could we use these to perhaps rescue the vascularization of tumors, or inject some flow so we could inject some anti-angiogenic drugs or so on. So we wanted to play with these intact tissues and their flow, because we wanted to see, if there's some remnant vascularization, obviously there's going to be some mode of drug delivery that's going to be very interesting. So we designed these little traps that have they're all connected to a single to a single. This is the size of a multi-well plate, and they're all connected to a single aspiration inlet. And we took these breast cancer cuboids from a mouse model called PY8119. I don't know why they named them like this, but there must be a reason. And then we put them in the traps, which is like a little well, like an open well, and we started applying suction. And this is actually a real cuboid after staining for the vasculature, and we saw very interesting results. So I'll go back to my laser pointer. So on the controls, after 2 hours, we saw some vasculature which going back to after 24 hours, that vasculature had almost disappeared. But but by keeping a perfusion of 0.1 ml per hour, that perfusion, even after 2 hours, we had kept it much. The vasculature had been maintained with little reduction over 24 hours. A little bit, more of a loss of 72 hours, but nothing compared to what you lose over in the controls, in which you see it almost totally disappearing. This is, by the way, this is very preliminary, and I have to thank our bright undergrad Brandon Nguyen and my postdoc Tran, but for doing this kind of in a hurry. But this is based on a very preliminary analysis of a very large number of cuboids. These have all been, each of these has been confocal imaged, and we haven't fully done a 3D analysis of it. So it's all based on a preliminary analysis of a projection, a 2D projection of these. So these are, I think, we'll see much more interesting stuff when we do a full 3D analysis of the vasculature. But even with this plain projection, I think the data already speaks by itself that you can maintain the vasculature by perfusing intact, which is fantastic. Anyways, going back to our cuboids, we wanted to see if you could see other, like the same question that we asked about about the slices. What did these have inside? We looked at immune cells, endothelial cells, proliferation, and this is a comparison between day 0 and day 2. We always want to look day 2, day 3, so that we could have time to do some drug testing, and we saw they were there. So this seemed to be like in the slices, we have a window in which we can do all these tests. And so we're very excited about this because, given the fact that people are all excited about organoids, we seem to have something that essentially looks like an organoid, but it is taken from patients, and it has the original cells of the patients that are intact, pristine. Nobody has ever touched them, and we can do all the analysis from those. So we started thinking about how to better make these platforms, and so this is all you see here is the work by my student Ethan Lockhart, who has done a lot of work with CNC milling, these platforms are very difficult to make at this. So again, it's very simple to make something small in CNC milling, but it's very difficult to make something reliable with small channels in the size of a 96 wall plate, with plastics and several layers of plastics that are bonded. And it turns out we needed valves to isolate the cuboids from each other, because we wanted to put different drugs, right? So they could, and because they're initially connected, since we have to load them, afterwards we had to disconnect them somehow. So we needed valves, and the valves are here. And he put together this beautiful video valves, and you can see that the valves are working because this is the meniscus of the fluid, and he's switching here from 1 Hertz to 2 Hertz to show us that they're working very well. So all these are working throughout a 96 well plate, and so he's done a fantastic work optimizing this process, which is actually very difficult to do that over such a large area. And we use a combination of hot press, a laminator, etc. And we'll be polishing this very soon. And I need my laser pointer back. So this is actually another mode of another way of immobilizing cuboids that we investigated in the past based on Takeuchi traps, which is interesting. We kind of abandoned this because it was difficult to make in large over large areas, but this allowed us to demonstrate that we could do some multi-well plates initially when we started this, and we did some Cytox green, cell tracker orange, cell tracker green, then Cytox green, so some playing with these, to show that we could do independent drug or dye type of exposures in cuboids. And the way it works is this is going back to the other design. I apologize for the confusion because we have done 2 or 3 designs, and it's important to always design this in a way that they are all very symmetrical. Otherwise, it's difficult to make them work. And so now we've grown that design to something bigger, and the way we see this is we try to see them all at the same time while we apply suction, and it doesn't work well in cell culture medium, and so we someone came, I think it was Lisa Horowitz in my lab, or someone in my lab came up with a solution that to add a more viscous solution like PEG temporarily to make these flow in a more reliable way, and it worked very well. So with 20% PEG. That's a 8,000 molecular weight. It's kind of like a loading solution. Then these flow very well into these. It's still not a perfectly reliable solution, and we're still going back and forth between one design and the other, and now we're still deciding what is the best solution. And I believe that the challenge is in doing it over very large areas. If you tell me, if you would be happy with something that is 1/4 of a 96 well plate, I would say that's a piece of cake. What is very difficult is to fill a 96 well plate with these tiny, with this. It's a reliability over large areas that is very difficult. But we're working on it, and we're getting better and better. These are just examples of things that are to show you how good we are getting at these, and so these are images that my students give me, and these are cuboids filled into the, and I don't see, I forgot what these were, but I think these were from a xenograft, and not from a patient. And and so, but what this shows is that each well, because they were the microvalves, we could fill independently the cuboids, the wells with different with different drugs and have different colors. I believe, yeah, this one is the 96 well plate. This is the latest generation, and we would have done this device was working. So except for the fact that for this particular isolation of the tumor, we did not have a full as many cuboids because the tumor was small. So, but anyways, it was working well. But not everything we do is microfluidic. I'll just rush through this because I don't have that much more time. Multi-plates can be very useful. Here we used 96 well plates to do real-time glow. It's a lifestyle viability indicator to show the sensitivity of the cuboids to a panel of kinase inhibitors. And you can note that the similarity of the signal from two duplicate wells. And as you know, cytokines can play very important roles in immunotherapy, cancer drug resistance mechanism, cancer cell progressions, and in many other physiological processes and signaling pathways that are very important for tumor development, tumorigenesis, and metastasis. So we measured the levels of 15 cytokines in the supernatant of the cuboids, and this was using Luminex, which is a bead-based cytokine assay. And so we found that numerous cytokines were secreted by cells in the cuboids, and this reflects the complexity of the cuboid microbiome, right? And so then we also did. Then we asked which cytokines might be increased by LPS stimulation. LPS is a molecule it's from bacteria that stimulates cells of the immune system. So this increase in the cytokines, it demonstrates the action of the immune system in the cuboid, in the immune tumor microenvironment in the cuboids. And so, and finally, I also asked my postdoc, Lisa Horowitz, to do something very painful, which is to manually seed cuboids in a 384 well plate. And so this is there's about 50 cuboids here per condition. And we wanted to look at the heterogeneity. So if you look at so there's a cytox screen is used as an indicator of cell death for cytosplastin and staurosporin, and noting here that we used 100 micromolar, and this is about 3 times or 8 times higher than in vivo. It's because we didn't know what we might be able to, what kind of signals we might be able to achieve. But using power, so the point of this was to do a power analysis, because we wanted to, this is the key for these and the slides that come now, that we determined that we would need 9 cuboids, we would need to test 9 cuboids per condition in order to detect a 30% increase from control with a false positive rate of 5% and a negative false positive rate of 20%. That's what these parameters mean. So you can change this if you want and get 10 negative a false negative rate of 10%, And then you get different numbers. And this is a very simplified analysis. We've learned from the Gervais group that you can do Monte Carlo simulations, and we're working on this as well. So you can change this and if you want to look at a 50% increase, for example, you might get you might only need 6 cuboids to get to that type of increase. So it depends on what is it that you're looking for. But that's what. So it depends on your goal or what you're looking for. But it's very important that you do this power analysis when you're looking at these. Okay, because essentially that you have some heterogeneity, and so. We also did it for the real-time glow, which looks, instead of cell death, it looks at viability. And here we determined, for example, that we would need 15 cuboids for if we wanted to see a 30% reduction. And because the data doesn't look as good as with cell death. And in the device, because there's more variability from sometimes there's multiple cuboids per trap, depending on how we're trapping. We're still working on it. These numbers don't look slightly bigger. So we would need between 20 and 60 cuboids per condition to obtain some of the levels that we want to see in reduction. When we look at this, these are by the way 3-day treatments, and the signal, the real-time glow, is analyzed between day 5 and day 2. So these and I'm only showing you the images from day 5. You can see notably that the viability at day 5 is much lower for cisplatin, but this is done at day. It is the ratio of these signals taken for each spot from day 5 to day 2. And just to finish, because this is my last slide, is something we're looking for the future, is that we want to do continuous monitoring of cytokine activities. Everything I've shown you, except for real-time glow, all these staining are the one-time shot, and we think that we can extract so much more information if we do continuous monitoring, that we're building these using dry film resist technology. Just essentially, it's a laminated SU8 that you use to pattern electrodes. We pattern the electrode, and it's important to do dry film resist because you need to pattern the electrodes on the top surface of the wells after you've fabricated the wells, and so that's shown here. And you need to fabricate 3 electrodes: the counter electrode, the working electrode, and the reference electrode. And I don't have time to go to electrochemistry, but this is based on aptamer technology. And we already have good working, we've detected cytochrome C already at acceptable levels in these, using these that my postdoc Tran Nguyen is fabricating. So to finish, I just want to say that I think microfluidic functional testing has enormous promise because for me, the tissue slices or cuboids, the 2 platforms have different merits, but they retain the tumor microenvironment. You can do multiplex drug testing, and I think it's important also to remember that this is by no means restricted to cancer. We are focusing on cancer because I think that that's where the questions are perhaps more important to health, but there's also a lot of other things that are out there where we would be happy to collaborate with these platforms. So I'm done. I was told that I would be able to allow for more for some questions for 5 or 10 minutes. I was also, I promised Ali that I would show just a few pictures of my bringing art into technology program, which is very popular. Everyone that walks, every student ever that comes into my office always asks me about my program. We've done a lot of exhibits and brochures. We have a YouTube channel. You're welcome to visit it. And these are just images, just examples of images, and that are very beautiful. I think just microfluidics alone is very beautiful. We do it because we think that art is something that images in general are something that are very self-explanatory, and when anyone can understand the beauty of art, and this is a way to get to approximate science to people through the beauty of art. And these are just images that people ask us, oh, what is this? It's so beautiful. And then it engages us into a conversation with them to make them, try to explain them what we do. So, yeah. Thank you very much. Done.
Fantastic talk, Albert. Truly appreciate it. With the art slices, a lot of questions came in, so let me just read them. Very impressive talk. In your 96 well system, does the liquid flow equally to all well when there's a lot of branching in the design?
Yes, it is designed that way. We've published the layout of the on the previous platforms and this platform. So we always make it always symmetrical, so that each channel has the same path to the outlet. Now, when you go to there's an exception to that rule, and it's that here, when you fill it, in this particular design, when you fill one, the resistance changes the moment one gets so the order in which they start getting filled changes. So that's where I think that the PEG makes a big difference between, so with normal cell culture medium, the difference from one when they start getting filled, it can give a lot of variability, because when you plug one end or the other, it makes a big difference. But with PEG, there's less difference.
Thank you. Can you go smaller than 96 wells?
I believe so. It's gonna be challenging. Depending on what design we use, is going to be challenging. So we've and it depends on what we use microfluidics for, whether we use it for loading or just for exchanging fluids. So I think that's going to be, the distribution lines for a 384 well plate can be a little daunting, and we're working on it. But I think we should solve first the 96 well plate. I think it is possible.
Thank you. Another question came in. How many days can you keep the tumoroids alive or functional?
Yeah, we typically go like 4 or 5 days, and I think I think it should be possible with a little bit more investigation. I think it should be possible to go for a week.
Okay, thank you. Very interesting talk. As I work with the 3D cell culture, I find it very challenging to sterilize the system. Also, type of cell media used for heterogeneous samples.
What was the question? The
person how do you sterilize the system? And also for a heterogeneous cell sample, each cell wants a different media. What type of cell media do you use?
I don't know the exact formulation because it's complicated, but it's not a very special. For example, for the colorectal liver metastasis, for example, I know that we use a specific one for that is given to us by our collaborators that we're using that optimized it for their tumor liver slices. So we changed that. It's not always the same one. And as for sterilization, yeah, we do as much as possible inside the tissue culture hood, but there's one step that presently we do outside, which is the cutting of the cuboids. And when we load the cuboids, right before loading the cuboids, the whole device, which has been sterilized with ethanol, then has to be loaded and perfused. So just the perfusion seems to be enough. And but it's a good question. I've always, been worried about that, and this does not seem to be a limiting problem. And I'll be happy to answer questions about these practical things, because my students know much more about this than I do. And but I'll be happy to put these people in touch with my students to know, because these are practical problems that I'm not very worried about them, because typically once we solve these are things that can be done inside the tissue culture hood, etc. So we're not at that stage yet, and we don't have sterility problems. My students solve them as they go, so to speak.
Thank you. Can you choose the size of the cuboids in the micro dissector?
That's a very good one. Yes, there are 3 cuts, right? So the first cut cuts the slice, and then the other 2 cuts are determine the other sides of the cube. So you can yes, you can determine, you can specify, you can decide how big you want them. Yeah.
Thank you. There's a concept in Industry 4.0 and continuous biomanufacturing of the digital twin, a mathematical clone of a given system. Can we see the organ-on-a-chip system as an organic twin of the system under investigation?
I'm afraid I don't understand the question.
The question is, I guess you're getting biopsy punches, other things. Can you make an organ-on-a-chip system with maybe spheroids to compare, your results and organ-on-a-chip results?
You mean instead of tumor on a chip, but with other organ tissues?
My understanding is you can take the cells, you can dissect them, put in a gel, and then print into different corners, and you can make a maybe 96 well different organoid sample rather than taking the native tissue from the cancer.
Yeah.
So can you compare, a human spheroid versus your real samples from the biopsy, and would they compare okay?
But ours are human. Ours are human. They're cut from a human. What would be the other comparison?
Same thing. You take the biopsy, you separate the cells, then make them a spheroid.
Oh.
Spray it into the well.
Well, I guess someone else will have to do that experiment because it's a lot of work, and I'm not gonna. Yes, I think it would be an interesting comparison, but I don't know the answer to that question.
Okay. The other one is, what are the percentages? What are the percentages of other cell types in your 3D tumor cuboid T cells, B cells, and macrophages?
The percentages. That is a very good question. We have not calculated it, and we did that. We did that more of in the cuboids. We have not calculated it. In the slices, we've done a more quantitative measurement, also over time, in a previous paper that we published in 2020. So, but sorry, we have not done that like a count of cells in the cuboids, but it is something that we should do, definitely. I agree.
Thank you. Another question came in about comparing CNC milling versus soft lithography. You pick CNC milling. What is the advantage over soft lithography?
Oh, manufacturing of plastics. It's very difficult to. It's very difficult to, and in general, if you want to bring that to a point where it's manufacturable in soft lithography, it would be very difficult to make these devices by soft lithography. You could not make this by molding, but in general, it's mostly about the materials. So we're interested in thermoplastic materials of well-known biocompatibility. Yeah.
Thank you. I guess you moved out of PDMS to PMMA for various reasons. Okay.
Yes, yeah, and mostly for manufacturing, because we cannot eventually, when we move out of the university and into a company, PDMS is very difficult to manufacture.
I see. Thank you. Great talk. How do you remove non-attached cuboids?
Well, we don't remove them from the device, but we started talking about. So they're not attached. They don't attach to a surface. But yeah, we started talking about how to take them out of the device for further analysis, like some genomic or proteomic analysis. And they're if they are embedded in a gel, for example, you can just aspirate them, and they're not attached, so they're not like cells or anything.
Thank you. Thank you. Another one is, what is the concentration of LPS in the system? Can you induce sepsis by injecting a high concentration of LPS and using your model rather than animal?
I don't know the concentration of LPS that was used in the experiments, but please email me and I'll be happy to I'll be happy to answer that question and put you in touch with the person who did the experiment.
Thank you. And final question, what is the minimum number of cuboids that you need to collect meaningful data in ELISA Luminex?
For a particular ELISA Luminex?
Measurements, yeah.
Yeah, for that particular assay, I don't know. We only started doing all these biostatistics analysis later on. We had not started doing it for the Luminex, but it should not be too difficult to go back to that data and do that analysis if that person needs us to do it. if they want to contact me. Yeah.
Thank you. Your sensing work, the final slide looked very interesting. Can you measure the level of different cytokines in cell media from your 3D culture over time for continuous measurements?
Yes, we believe so. Yeah, I mean, that's what we're working on with the electrochemical measurements. Yes.
Okay, awesome. Well, that's all the questions I have, Albert. Thank you for your time, for a great, fascinating talk with artistic slides, very beautiful slides. Thank you so much.
Welcome. Thank you for having me.
Have a nice day. Have a nice day, everyone.